Greetings and welcome to the 2026 National Invasive Species Awareness Week webinar series. My name is Tegan Wilmot and I serve as your Education and Prevention Manager at NISAW. Please say hello in the chat and share with us where you are joining from today. Thank you again for helping kick off National Invasive Species Awareness Week with our first webinar, USGS Invasive Species Science, Innovation for a Healthy Nation. We are excited to once again be hosting National Invasive Species Awareness Week, an international event powered by NISAW that brings attention to invasive species, the impacts they have across North America, and the actions we can take to prevent their spread. This year's theme is Defending Our Health by Stopping the Spread. The theme highlights the many ways invasive species affect not only ecosystems, but also human health and economic stability, including impacts to agriculture, infrastructure, recreation, and community well-being. It also emphasizes the importance of proactive prevention, early detection, and coordinated response to reduce these risks. NISAW provides a platform for people from local, state, federal, tribal, and regional agencies, nonprofit organizations, private industry, academia, and concerned citizens to come together and engage with decision makers around legislation, policy needs, and on-the-ground solutions. This week is designed to support both learning and action. Throughout the week, we'll be sharing links and resources to help you connect with your U.S. senators and House representatives. We want elected officials to hear directly from practitioners, partners, and community members about why invasive species prevention and management matter and why sustained support and funding are essential. For those who want to take a more active role during NISAW, we encourage you to visit NISMA.org and explore the NISAW Toolkit, which includes template social media posts, press releases, and outreach materials. If you are not already connected with NISMA on social media, please follow along and help amplify our daily messages. We have a full week of webinars planned featuring presentations on topics such as Alberta's wild boar control program, New World screwworm efforts in Mexico, marine biosecurity and public health, and the connections between invasive species and food security in the United States. We encourage you to visit our website to view the full schedule and register for additional sessions. If you are unable to attend a webinar live, recordings will be posted to our YouTube channel following the event. And before we welcome our speakers today, I'd like to take a moment to share a brief overview of NISMA for those who may be new to our organization. NISMA, the North American Invasive Species Management Association, is a nonprofit organization, and our mission is to empower invasive species management in North America. We serve professionals, practitioners, and researchers working across disciplines to prevent, detect, and manage invasive species. NISMA is guided by 16 board members and supported by 10 active committees that help shape and strengthen our programs. We have a team of 7 staff members and a membership that continues to grow each year. We currently communicate with over 15,000 subscribers and engage with over 38,000 social media followers. During National Invasive Species Awareness Week last year, our collective outreach reached more than 3.4 million people, and we are excited to continue expanding that impact this year. What do we do? Well, at NISMA, we provide the connection, the voice, and the tools needed to address invasive species across North America. Our work is organized around these 3 core pillars. We create opportunities for collaboration and knowledge exchange through webinars, summits, our annual conference, and professional discussion boards. Our goal is to connect practitioners, researchers, policymakers, and partners so they can learn from one another and strengthen their impact. We elevate invasive species issues on a national stage through our government relations program and initiatives like National Invasive Species Awareness Week. And we work to ensure that decision makers understand the importance of this work and the need for sustained support and funding. We equip our members and partners with practical resources for prevention, management, and outreach. This includes Play Clean Go, the NISMA Shop, the Certified Weed-Free Products Program, Mapping Data Standards, and NISMA Academy. As we continue celebrating National Invasive Species Awareness Week, I also want to quickly highlight the NISMA shop at shop.nisma.org. You'll find root brush stations and replacement parts, buckthorn blasters, apparel, outreach materials, and more. These are practical tools that support prevention and management efforts across North America. In honor of National Invasive Species Awareness Week, you can use code NISAW26 for 10% off your purchase. Members can stack their member discount on top of that, and every purchase supports NASMA's programming. This promotion is available this week only, so be sure to take advantage of it while it lasts. One of the most important ways we facilitate connection is through our NASMA annual conference. Each year, over 300 invasive species professionals gather to share research, case studies, and management experiences. This year's conference will be held in Alexandria, Virginia from October 19th through the 22nd with the theme Local Leadership, Global Impact. We have some great keynote speakers lined up already, and registration is now open, and we're also accepting abstracts. You can learn more on our website. As a friendly reminder, your membership dues help to fund our educational programs like this webinar. If you are not currently a member, we offer a variety of different membership options. You can view a membership structure on our website, nasma.org, and if you're unable to become a member right now, we encourage you to make a donation. Every gift, no matter the size, helps to support our programming. One of our newest member benefits is our NASMA discussion boards. When you become a member, you'll have access to these boards where you can ask your peers questions, share resources, discuss emerging challenges, and learn from peers across different sectors. Is someone else trying to manage the same species as you? Are there new innovative solutions that might help you solve a problem? This is a great place to start. We also have invasive species news, job opportunities, and trainings listed too. And we're really excited about this new member benefit and hope you will join us on the discussion boards soon. If you want to sponsor NASMA in a bigger way, our Premier Partner Program is designed for you. Premier Partners enjoy benefits throughout the year, including acknowledgement on our website, at events, and more. And we'd like to acknowledge and thank all of our 2026 Premier Partners whose generosity helps to make events like this webinar possible. And now we'll get started with our main event. Thank you for joining us today for our first NISAW webinar, USGS Invasive Species Science, Innovation for a Healthy Nation. Before we begin, a quick note for our audience. Please put your questions in the Q&A box so we can make sure to address them after the presentations. Take a look first to see if your question has already been asked, and if so, give it a thumbs up to promote it to the top of the list. To get us started, I'd like to welcome Paul Heimowitz, who will be moderating today's webinar. Paul is the Terrestrial Invasive Species Program Manager within the Biological Threats and Invasive Species Program of the Ecosystems Mission Area. He coordinates research on prevention, detection, monitoring, control, and other management priorities, addressing a wide range of terrestrial plants and animals. Paul, thank you so much for joining us today. We have a great lineup of presentations, and I'm excited to get started.
Thank you so much, Tegan, and thanks everyone for joining today. A huge amount of appreciation to NASMA overall for hosting today's webinar and their leadership in National Invasive Species Awareness Week. As Tegan mentioned, I manage our invasive species program within the Biological Threats and Invasive Species program. And I'm also here today with the webinar co-organizer, James English, who is our biosurveillance network coordinator in the same program within the USGS ecosystem mission area. We really appreciate the opportunity today to showcase just a sampling of how USGS invasive species science aligns with this year's NISAW theme of defending our health by stopping the spread. So before I introduce our lineup of presenters, I just wanted to briefly note that USGS has a long-standing program and role producing scientific products that help natural resource managers prevent, detect, and respond to biological invasions across the country. From our brown tree snake biosecurity research and rapid response team in the Pacific Islands, to development of new detection and control tools for aquatic plants and animals invading the Great Lakes, and many other efforts. Our scientists collaborate across our many science centers and with numerous research partners. This slide summarizes our areas of emphasis with just a few examples of some other areas that we work on besides what you'll hear about today. And I'll put a link in the chat in a moment to a recent fact sheet we produced that provides a more complete overview of the breadth of our invasive species science. Today, you'll hear from 5 stellar USGS scientists working on a range of facets of our innovation for a healthy nation. Dr. Camille Hopkins, a wildlife disease specialist and another member of our Biological Threats Headquarters program, will set the stage with a talk on the role of invasion biology in a One Health approach. Next, Dr. Katie Klimas with the Columbia Environmental Research Center will talk about development of point-of-use environmental DNA tools for invasive species, parasite, and pathogen surveillance. After Katie, Dr. Heather Walsh with the USGS Eastern Ecological Science Center will present on heavy metals, PFAS, and other health effects of invasive blue catfish in the Patuxent River. Dr. Michaela Phillips with our Pacific Islands Ecosystem Research Center will speak on bridging science and practice to reduce fire risk from invasive vegetation in Hawaii. And batting cleanup, Dr. Mary Beth Bray with the Upper Midwest Environmental Sciences Center will present on developing acoustic deterrents for invasive carp control. With no further ado, I'll turn the mic over to Camille and we'll get started on our presentations.
Paul, thank you so much. And thanks to the organizers and everyone for being here today. Happy National Invasive Species Awareness Week. Okay, so first of all, I wanted to start with this picture of New World screwworm, an invasive, well, maybe invasive, right, depending on how you think about it, fly that we're concerned about coming to the north into the United States, back into the US from the south. And this issue is one where I have personally been advocating that we need invasive species biologists and experts to be engaged. And today I want to give an example of the link between invasion biology and One Health by considering specifically how invasive species can impact One Health sectors. So what is One Health? It's an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. And the reality is, when we think of these different sectors, we have to consider invasive species in each sector because of the role that they can play in the dynamics of disease. One other thing I want to point out is from the level of how do we manage threats, emerging threats to health, we need to include invasive species expertise in the 4 C's of One Health: communication, coordination, collaboration, and capacity building. And One Health is not a new concept. If you have enjoyed the readings of Aldo Leopold, as I have over the years, his concept of the land ethic really is what One Health is all about, that we have to consider the relationships between all of these things. And that's something that Indigenous communities globally have also recognized. So today I want to consider the symbiosis of invasion success, invasive species, and the spread of disease, and specifically the ideas of co-invasions and niche construction. So I'll start with an example of a disease, rat lungworm, a disease that can affect people that is spread by giant African land snails. And in partnership with the state, with CDC and others, USGS scientists investigated this years ago in Florida, finding that in 6 of the 7 core areas where rat lungworms are found in the greater Miami area, there was detection of this lungworm. So here, the rat lungworm has benefited from the invasion success of this snail. Similarly, as we look to the south and consider how with changes in the environment, we might see vampire bats spread north into the US, we have to acknowledge that they carry a different type of rabies virus than what we have been living with here in the US. And a well-known invasive species threatening Florida, the Burmese pythons, we know that they can actually play a role in spreading a parasite that is unfortunately negatively impacting native snake species in Florida. The other piece that we have to consider is that through changes in the environment, it's possible for invasive species to create a niche, an area where they can proliferate and disease can proliferate. The first environmental change I want to highlight, we're seeing more of this in our world, increasing frequency of storms and disasters. And if we think about dust storms, for example, and these are things that where you see in this image, theoretically, we know that changes to the environment have the potential to affect disease dynamics by affecting species composition and diversity. And so with dust storms, it's been shown that dust storms as far away as Mali can spread fungal pathogens, invasive pathogens to the Atlantic, impacting important species like coral reefs. In my own research, in my PhD, I found that as we change the structure of our forest through changes through fragmentation, for example, we actually are creating an environment that allows invasive mosquitoes to persist and pick up our native diseases and spread them. The last key thing I want to leave you with is the idea of biological threat squared. The idea that as we think about how do we manage invasive species, how do we manage disease and threats to health, that we need to think about them together. And in order to do so, it's number one, thinking about the enemy release hypothesis, parasites or pathogens left behind. Number two, thinking about how we can align the approaches that we use in dealing with invasive species and disease. And then finally, how can we leverage innovative tools that can help for early detection of not only invasive species, but also for surveillance of disease. And so the enemy release hypothesis first highlighted in Darwin's Origin of Species, but then later really developed by Elton in the Ecology of Invasions, recognizes that these species have their enemies. Oftentimes we're thinking predators that regulate their populations in their native environment. But if they invade into new spaces, those enemies might be reduced or absent. And the reality is parasites and pathogens are also enemies. And we should think of the enemy release hypothesis in that way. A study from 2003 that included USGS scientists found that European starling, for example, carry in their native range 20-something species of parasites, and in their introduced range, that has been reduced to 9. Similarly, with black rats, it's been shown that they only retain 38% of their native parasites in their introduced range. Similarly, with European green crab, certainly a hot topic right now, it has been shown that they also get release from their parasite enemies, allowing them to spread globally. And in this image, what you can see to the right, the green shows you the introduced range, and on the y-axis, the parasites compared to the native range, and you can see a significant reduction. The reality is parasites and pathogens help regulate populations, and that is really critical for us to understand if we want to manage invasive species and control disease. And this type of approach, thinking about biological threats squared, is something that we do in USGS under the Ecosystems Mission Area Biological Threats and Invasive Species Research Program. Together, our program looks at disease and invasive species. And as Paul already mentioned, we have similar sublines of work for our research. And that science, working together, we are able to consider these threats to our ecosystems, but also threats that can impact the agricultural economy and public health. Across the nation, we have scientists working on wildlife health issues, looking at what causes death and illness in wildlife populations across the landscape or aquatically. And USGS has that unique niche of thinking about not just are there parasites and pathogens present, or are there invasive species present, but combining that with long-term ecological data and earth data to understand the ecological dimensions of diseases and invasion. And that can provide us with actionable data to develop tools and strategies for natural resource managers. We acknowledge that parasites and pathogens are part of the ecosystem, but there are many that can significantly impact populations and ecological communities. And one of the fun things that we're looking at in terms of innovation, bringing together tools, we are building off of the Non-Indigenous Aquatic Species Database, NAS, which many of you know, and we're putting on top of that infrastructure a repository to look at aquatic diseases and pathogens. We're also looking at innovative tools like environmental DNA, and in fact, looking at robotic sampling that can benefit early detection of invasive species, but also parasites. And the key thing is, by understanding better the ecology and the invasion biology of invasive species and diseases, we can help identify ecological countermeasures that are going to promote the health of wildlife, as well as any diseases that might spread to people, pets, or livestock. So in the end, when we talk about One Health, the reality is we're only as healthy as our ecosystems. And I encourage all of you to check out our QR code here to learn more about One Health and USGS, but also to look into the Biothreats program and some of the great things that we're doing together to sort of think outside of the box for these wicked problems of invasion biology, invasive species, and disease. Thank you so much for your attention, and I look forward to any questions.
Thanks so much, Camille. That was a great overview, and lots of well-deserved virtual applause coming your way. I don't see questions in the Q&A yet. Folks might be still warming up, but I know, Camille, there's so much more that your talk made me think about. I wonder if, while we might be waiting for other folks to put in questions, you could just talk a little bit more about kind of your perception or from the wildlife health realm in general, the distinction that you noted as well is sometimes tricky to make between invasive and pathogens and parasites and New World screwworm as kind of this tricky example. Yeah, it'd be interesting to hear some more perspective on that.
I'll just highlight for folks in the audience, if you didn't know, that New World screwworm actually was a native of the Southwest United States, but it spread invasively to the Southeast US and had a significant negative impact, not only on livestock, but also on wildlife at that time. And then it was USDA who in the 50s and 60s developed some novel techniques, the sterile insect technique, to push New World screwworm down to the South and eradicate it from the United States. And then in partnership with Mexico and other countries to the South, by the early 2000s, New World screwworm was actually eradicated or controlled all the way down to Panama and the Darien Gap. Unfortunately, we have seen some changes in the landscape, and this is Camille's hypothesis, but because there have been some changes to the landscape, I think that is what allowed New World screwworm to overcome the barrier of the wilderness of the Darien Gap and began to start spreading north. There's also some theory, some hypotheses about maybe some resistance to some of the techniques that have been used to control it. So is it an invasive coming into the US, or is it a native returning back to its original habitat? So definitely would love to hear from folks in the chat on what you think about that.
Thanks, Camille. That was a great answer. There's a question in the Q&A that I think we can answer a little more quickly, just asking about some website resources. So I'll share one that came in the regular chat you might want to talk about. Question is, the person notes they're curious to hear more about ecological countermeasures, and if any have been considered recently. They said, I have heard about introduction of parasitic species to control insects in agriculture, for example.
So I'll say for ecological countermeasures, first starting from what I know regarding wildlife health, one of the things we point out is that with highly pathogenic avian influenza, bird flu, and certainly some of those new strains are invasive to the US, we have seen that there is an interface between wild birds, primarily ducks, waterfowl, and agriculture. Our scientists at USGS actually, before it was detected in dairy cattle, published to say that waterfowl are spending time on dairy operations. And so the reality is, but this needs to still be scientifically proven with some more data, it seems as though if we can make our natural habitats more appealing, in other words, restore them, reduce the fragmentation of wetlands, is there an opportunity to then maybe reduce that interface between wild birds and agricultural areas? So that's one potential example. The other, in terms of ecological countermeasures that's been shown globally, where that publication came from, related to some viruses that are carried by flying foxes in other parts of the world, where it's clear that if we can restore some of the native habitat of these flying foxes, they will be less likely to come into closer contact with domesticated animals and spread these horrible diseases like Hendra virus and Nipah virus.
All right, thanks so much, Camille, for getting us off to a great start. I think we'll need to move on now. So if folks have other questions for Camille, feel free to continue to put them in the Q&A. And there's some general questions in there we'll get to as well. But at this point, I'll go ahead and move the baton on to Dr. Katie Klimas. And Katie, let you take it away.
Okay, thank you. All right, well, thanks everyone for joining. I am super excited to talk to you today. I'm going to be talking about the work and research that the USGS Ecosystems Mission Area is doing on developing molecular point of use tools for the detection of invasive species, parasites, and pathogens. A large portion of this work I'll be telling you about today is actually being conducted by Steve Spear and his group at the Upper Midwest Environmental Science Center, and the work that they're developing these tools for invasive species monitoring and pathogens. And then at the end, I'm going to wrap it up with a project that Steve and our group at the Columbia Environmental Research Center is doing on developing these tools for the New World screwworm. Okay, so hopefully some of you guys are familiar with environmental DNA and environmental DNA techniques, but just really quick, it's, taking a water sample, a soil sample, extracting DNA from that, using methods such as polymerase chain reaction to amplify that DNA, then using tools such as quantitative PCR or sequencing to be able to visualize that DNA and say, hey, I've got DNA of this species, maybe, in this lake. So point of use molecular tools are a little bit different in that rather than having to go through that very extensive process that can take a lot of time, they allow for on-site and rapid detection of an organism's DNA or other biomolecular marker. And so they don't necessarily require the complex laboratory equipment and infrastructure or training. And these methods and techniques have been used in medical diagnostics for quite a while. So I'm sure a lot of us are familiar with the COVID-2 strip tests that we probably used in the past. They've also been used for HIV, Zika, tuberculosis, and malaria. And the concept here is that we can take a sample and determine whether a particular biomolecular marker is in that sample very rapidly, within a few minutes. And so what Steve and his group have been doing have been to try and take these techniques and use them for invasive species detection with environmental DNA. So some important characteristics of these point of use molecular tools include a rapid time to results. They require minimal training, but you still want fairly good sensitivity and specificity of your target. And a lot of these characteristics are very useful for invasive species applications. So, the portability of these tests allow for surveillance at various places, at points of entry, or really anywhere on the landscape. They allow for the detection of these targets at any place in the field, potentially. They allow for increased ability in monitoring events, and also particularly where management is time sensitive. So if managers would like to get an answer fairly rapidly within an hour, these types of tools might be able to do that. And finally, they also provide a lot of flexibility for the field staff. So these tools do not require a lot of the extensive laboratory infrastructure that's needed for our usual eDNA workflow. So I'm going to just walk really quickly. This is our typical eDNA workflow. So we've got field components and we've got lab components. So first in the field, we take our sample, and then we take that sample back into the laboratory. And so we have to do a DNA extraction. And our usual DNA extraction protocols require high-speed centrifuges, multiple reagents, and a lot of different steps. And so they take a bit of time. Once we get that DNA extracted, we can then run our assay, so our qPCR assay. And doing that requires a lot of reagents, and these reagents require refrigeration generally. Once we get that assay prepared, we then run it on our thermocyclers. These are instruments that require increase and decrease in temperature. So they need to cycle through various different temperatures. And they also need a fluorometer for detection of that DNA. And running these samples usually can take 1 to 2 hours for that analysis. Once you get those results, you can then communicate them back into the field to the manager. And I just want to note here that a detection of eDNA does not necessarily result in an immediate, boots on the ground action, but it could entail a management decision of taking more samples. So contrasting that with the point of use workflow, everything can actually be done in the field. So we can collect our sample, take it to this portable, basically a heating block, and run a DNA extraction. We can then use various techniques that I'll be talking about, including a LAMP assay with stable reagents. So again, everything can be conducted in the field. And then these assays allow for simple results interpretation. So they can either lead to a color change or this lateral flow strip here, similar to the COVID tests that you might be familiar with. So you can get results quickly and really easy to interpret. And so then that information can go to the wildlife manager. Okay, so kind of went through that different workflow. And so now I'm going to talk about the work that Steve's group has been doing on taking these parts of that workflow. So kind of going back here. So the extraction portion, the testing portion, and the results portion. And what they've been doing so far to be able to get these into the field. So out of the lab and into the field. So for the DNA extraction, they tested a couple of different rapid extraction protocols. These have been developed for, some of these have been developed for a long time. Some have been a little bit more recently, especially to support rapid disease testing. So they tested 4 different methods, and they're classified into 2 categories. One that requires a heating step. So the Helix resin and the quick extract are solutions that just require a heating block and then release the DNA. These are great for moving into the fields. They don't require a lot of equipment. They're really rapid, but they don't necessarily have the cleanest DNA coming out. So you'll get a lot of cellular material in your sample still. The next group of extraction methods that they tested were syringe column kits. And these are very similar concept to the solid phase extraction kits that we generally use in the laboratory, but they're smaller. And so you do get a slightly purer DNA extraction, but they also require several steps. And they found that these filters can easily get clogged and they are a little bit more expensive. So basically Steve's lab designed an experiment to test these different methods. And I'm going to kind of walk you guys through their experimental design really quick. So there's a couple different phases. They took water samples from aquaculture facility as well as from a river. And that was in order to get background eDNA. So they have this DNA from a background, fish that were in the water. They then filtered them. Now I've got 2 different filters that they use, and that's really not important for this part of the discussion. So we're not really going to focus on that. But once they got the background DNA filtered, they then spiked those filters with target DNA, which were grass carp cells. So they know that they actually got grass carp DNA, which was their target, onto those filters. And then they extracted them using one of 5 different techniques. So the IBI is the control. That's the technique that we generally use in the lab. Then you've got those 2 heating methods, the quick extract and the Helix. And then the 2 syringe methods, the biomeme and the POC. After they extracted the DNA, they then used qPCR to amplify that DNA to see which extraction gave the highest concentration. And so these are the results, and I'm going to help walk you guys through them. There's a lot going on here, but we've got, again, we've got the 2 different phases, the mesocosm versus the river. So we're just trying to go from a more controlled setting to a more, realistic setting. The important part, though, is to look at the copies per reaction on the y-axis and the x-axis, which shows the different extraction types. The different colors represent the different filter types, but we're really not too concerned by that. So we've got the IBI lab method. We've got the 2 syringes, the BM and the POC, and then the QE and the CH are our heated extraction methods. And what I just really want to point out here is they found that the heated extraction solutions method, either the Helix or the quick extract, produce the highest DNA concentrations. So they decided to take those and move forward with the rest of their work to try and move these protocols into the field. So now that we've got our DNA extraction that we could do in the field, we then move to the analysis part. So normally we use PCR polymerase chain reaction to amplify that DNA. But what Steve and his group have been doing is using a different method called LAMP, or loop-mediated isothermal amplification. This amplifies the DNA without the need of a thermocycler. So you don't need to increase or decrease temperatures. You can just use one temperature, so you just need a simple portable heating device. You also get results much more rapidly, from 10 to about 40 minutes. But generally, the work that Steve's group has found is that within 10 minutes, you can get a result. And LAMP has been demonstrated successfully in point-of-care diagnostics, including Ebola and COVID as well. And so this is not necessarily a new technique, but it's already a fairly standard technique for medical diagnostics. One thing to mention though with LAMP is that it is a little bit lower sensitivity than qPCR. Okay, so one final step with getting these assays into the field is the reagents. So a lot of these reagents require refrigeration, various handling, but one way to get around that is through lyophilization. And so just basically by freeze drying the reagents, and then we can take them out into the field and run these assays. And so they took their zebra mussel assay and they lyophilized it. And so the results here are showing either the use of this assay with non-lyophilized reagents versus lyophilized. And the important part here is to just see we've got each of these lines that are different colors represents a different amount of DNA, of target DNA. And what you'll notice is that when those lines go up, that indicates an amplification. And so all the targets are amplifying as expected. There is a little bit difference in the RFU, but that's really not what's important. What's important is just the fact that they're amplifying and that the exponential phase of that line occurs roughly around the same time. So if you look at the x-axis, the time, between 5 to 10 minutes, they get amplification with either the non-lyophilized or the lyophilized. So now we know that we can take these reagents, lyophilize them, so we can take them into the field. We can run our samples that we extracted with a portable extraction method, and then use LAMP to test them in the field. All right. And then the one last part here is the simple results interpretation. So LAMP is able to give you either a colorimetric response, so you can get a difference in color of your samples for amplification or non-amplification. And it can also be used in these lateral flow strip, kind of like the COVID tests. And so I will be talking a little bit more about that towards the end, but I want to go through some of the these point of use examples that Steve's group has already been putting into action. So a number of aquatic invasive species they've designed these LAMP assays for, including zebra and quagga mussels. So they're using eDNA samples in Western lakes, as well as in the upper Midwest, they're doing toe samples and using these LAMP assays for quagga and zebra mussel. They have been developing a rapid assay for New Zealand mud snail, as well as spiny water flea. And finally, they're validating some LAMP assays for the 4 different invasive carp species. Finally, they're also using these on aquatic pathogens, including the largemouth bass ranavirus and viral hemorrhagic septicemia virus. They are also moving into using these point of use methods for terrestrial invasive species, including priority species in the Pacific Islands, such as the brown tree snake, little fire ants, and the great banded hornet. And finally, what I'll be talking about today is work that we're helping them with on developing these point of use assays and techniques for the New World screwworm diagnostic test. So Camille just briefly kind of went over a little bit about New World screwworm. So let me just kind of give you a little bit more background on that. As she was mentioning, it is native to North America, but it's a major agricultural pest and wildlife health concern. This is probably the second grossest picture I could find. I'm sure there's plenty of gross pictures out there on this, but it affects not just cattle, which is certainly the main concern for agriculture, but also wildlife. As Camille mentioned, it was initially eradicated down to the Darien Gap, so that little gap between Panama and the rest of South America, through the sterile male program that USDA implemented from the 1950s through the present. It's still ongoing. But as Camille mentioned, the past decade, the New World screwworm has been moving north and reinvading through Central America into Mexico. And so there's a need for more effective border surveillance for the New World screwworm. And so that's where our groups are coming into play. We are planning on developing a point of use tool for New World screwworm. So current surveillance for the New World screwworm occurs through fly traps and livestock inspections at ports of entry. But there are several similar fly species that are very difficult to distinguish and require expert taxonomic identification. And so if we can design a LAMP assay that would provide an immediate genetic identification with subsequent laboratory confirmation, we feel that that might really be beneficial for surveillance of this parasite. And so we are working together on designing a LAMP assay along with the DNA extraction, rapid extraction methods, et cetera, that Steve's lab has already demonstrated. We envision that we would be able to potentially collect DNA through wound swabbing at these ports of entry, as well as collecting adult samples from fly traps, et cetera. And then the end goal would be to design this test with, again, a lateral flow assay, similar to, again, the COVID test for really quick and easy to understand results. And with that, I'm going to wrap it up. So that is our project, and we're going to be working on that the next few months. So stay tuned for hopefully some exciting findings. And with that, I will thank our funders. Again, I've got Steve's email here and my email if you'd like to get in touch with us. And with that, I will take any questions. Thank you.
Thank you, Katie. That was excellent. And it's just so exciting to see how these tools continue to develop and with this new application for the scary New World screwworm problem. I think, we have one question in the chat that's kind of more general for screwworm detection that Camille, maybe you might want to answer, or someone else offline. But there's a question, Katie, we have just a minute, you might be able to touch on, which asks, can eDNA sampling be used to detect aquatic invasive weeds, such as submerged water milfoils?
That's a really good question. So my expertise isn't in plant eDNA, but I know a lot of folks that are working on plant eDNA. I think it's a bit trickier. There might be methods to help improve the detection probability with perhaps different ways of sampling and things like that. But the overall methods, yes, they are being used for aquatic invasive plants.
All right, thanks, Katie. Feel free to add other questions in the Q&A for Katie if you have them, but we will need to move on. So thanks again, Katie. Our next presenter is Dr. Heather Walsh. And so Heather, feel free to come on and share your screen.
Thank you everyone for joining. I'm going to talk today about heavy metals, PFAS, and health effects in blue catfish from the Patuxent River in Maryland. If you're not familiar with this area, the Patuxent River is considered a major tributary of the Chesapeake Bay. And I am a research fish biologist at the Eastern Ecological Science Center in Kearneysville, West Virginia. And I want to thank my collaborators on this project from Maryland DNR and also within the USGS. Andrea Tokunov and Zach Hopkins did PFAS analysis, and then a postdoc student from Morgan State University also analyzed microplastics, which I'll get to. So just to give everyone a brief background on blue catfish, they are native to the Mississippi, Missouri, and the Ohio River basins, but they were introduced by the Virginia Department of Wildlife Resources in the mid-1970s in order to create a recreational trophy fishery. Unfortunately, blue catfish were able to tolerate higher salinity values than what was anticipated. When Virginia DWR stocked these fish, they did not anticipate them being able to move throughout the Chesapeake Bay into other tributaries. The problem with these fish is that they like to eat and they grow big really fast. If you've heard about the issues associated with them, they're eating blue crabs, menhaden, striped bass, the various types of shad in the Chesapeake Bay. And these are all species that have been heavily managed for in order to try and increase populations. And so having this opportunistic predator invasive species in the Chesapeake Bay is really bad for these species. They're also outcompeting native species, and in some of these major tributaries are constituting up to 70% of the total fish biomass. This map is from the National Wildlife Foundation, and it shows all the tributaries that they have spread to from their initial stocking locations where the green triangles are. So at this point, they're basically in every major tributary of the Chesapeake Bay. As I've mentioned, they grow large and they grow fast. Females are capable of laying 4,000 to 8,000 eggs per kilogram of body weight. So I have in the bottom right-hand corner the definition of an invasive species, and that's the ability to rapidly establish, reproduce, and spread, outcompeting native species due to a lack of natural predators. And blue catfish have become very good at this. So I'm going to get into some of my data now. We've performed blue catfish health assessments in the Patuxent River now in June of 2024 and in 2025, where we sampled with Maryland DNR at 5 locations shown on this map. Western Branch site being the northernmost location and Hunting Creek site being the southernmost location on the river. Any further south, we were running into salinity issues and where the electroshocking boat just wasn't capable of shocking up fish. So at each of these sites, we aim to collect about 10 blue catfish and a water sample for PFAS analysis. So in 2024, we were able to collect 50 fish, and in 25, we collected 44 fish. And I'll just say that the data I'm going to present is from the 2024 sampling. Our health assessments are very comprehensive. As I mentioned, the blue catfish were collected by Maryland DNR. We weigh and measure the fish. We do an external assessment of any abnormalities that we see, including skin lesions, and we also assess internally for any changes that we observe. And then we also are able to calculate condition factor. We weigh the liver and gonad for gonadosomatic and hepatosomatic indices, measures of reproduction and energy. And then we also take tissues for histology to look microscopically at any changes that we can see. And those tissues include the gill, liver, spleen, head and hind kidney, gonad, and any other abnormalities. For this analysis, we tested heavy metals from the muscle. And then, as you can see here, there's a list of 10 heavy metals that were tested. And then any fish where we saw higher levels of arsenic, we also tested for inorganic arsenic, because that's the form of arsenic that's more harmful for human health. And as I mentioned, the postdoc from Morgan State University she only analyzed 5 fish for microplastics because we weren't even sure if she would be able to detect any. Not yet done is the gene expression analysis in the liver, and I'll be able to get that done once some of our funding restrictions are eased up. But the plasma was analyzed for PFAS, and we aged the fish with otoliths. So jumping right into the histopathology results, I just grouped all the different types of histopathological abnormalities together for this graphic. Length are the orange bars, weight is indicated by the gray bars, and then the number of abnormalities summed at each site is represented by the black line. So the highest number of abnormalities was detected at the Hunting Creek site. But I'll say that blue catfish, in addition to being voracious predators, they're also very good at swimming and they move. They don't just stay in one location. So it could be that these fish are moving up and down this stretch of the river. And so there's some overlap. You know, the ones that at Hunting Creek might have been at Hall Creek the week before. So just keep that in mind. The heavy metal results showed that arsenic was highest at King's Landing and was actually significantly higher than at Western Branch and Hunting Creek. And I have the sites ordered in the location from northernmost to the southernmost sites. Copper didn't really show, although there were significant differences. They were minute and was pretty steady across the sites. Iron was highest at Hunting Creek, and then zinc was highest at Lower Jug Bay. And as I mentioned early on, there were 10 types of heavy metals detected. There was a fair number of them that were not detected or were very close to below detection. So from 9 fish, I had inorganic arsenic tested, and I based sending those samples out due to consumption. There's not really a true consumption advisory for arsenic. There's recommended amounts that you should eat based on body weight. So just looking at the organic arsenic levels and that recommendation by the World Health Organization, I found these 9 fish that could potentially be have elevated levels of inorganic arsenic. And so the bottom there are each individual fish. Most of them came from King's Landing, and I'm going to go back to the last slide to look at arsenic, and that's where we had the highest levels of arsenic, so that makes sense. What I will say that I don't have all the data here of other species, but inorganic arsenic levels tended to be on the low end when you compare these samples to other fish published in the literature, other levels of inorganic arsenic in fish published in the literature. Then moving on to mercury and methylmercury. Again, we found highest levels at Hunting Creek, the most downstream site, but there was really no significant differences between the sites. Then moving on to looking at the contaminants and relating them to the biological endpoints we observed with both out in the field and with histology. So the list here in bullet points shows the 10 heavy metals we had analyzed and the range, and the ones that we did not really detect in any of the fish. What I really want everyone to get from this table, though, is that the only contaminants, mercury and methylmercury, those were the only 2 that were associated with size. So when it comes to fish consumption advisories, they generally give you a size that's safe to eat, but contaminants such as arsenic and iron and copper were not correlated to size. So if you're eating fish that are smaller, that are considered safe to eat based on mercury advisories, you still might be getting elevated levels of other contaminants that you might not be aware of. Most associations with arsenic were negative. It's interesting that arsenic had some associations with liver endpoints and posterior kidney. Posterior kidney is where generally a lot of the excretion part of digestion happens in fish, as opposed to the head kidney, which has more of an immune function role. Many studies have shown that mercury and methylmercury are associated with size of the fish, and that's what we're seeing here too: size, gonad weight, condition factor, and reproductive stage. Although we did see some associations with some of the gill observations, histological observations. And then copper, we saw some positive associations with gonad size. Although this seems like maybe it would be a good thing, it could be that this is just related to short-term exposure scenarios. Generally, what's seen in fish is that copper can inhibit growth and reduce long-term reproductive success. And if that's the case, that might be a good thing to combat invasive catfish, but it's kind of early on to say that at this point. There were 8 types of PFAS detected in every blue catfish. So that, like I said, this is just the 2024 samples, so that's 50 fish. That was kind of interesting to me. The other data that we have available that we've analyzed is from smallmouth bass. And generally what we've seen in them is that what we found, depending on location, anywhere from 1 to 5 PFAS detected in every fish. So there's a lot more being detected in blue catfish with the caveat being that the levels were much lower than what we've seen in species like smallmouth bass. It's too early to say why that might be the case. These catfish do seem to have a high metabolism, and it could be that they're metabolizing these compounds and excreting them faster than we can detect them. It could be their diet, and just it's hard to say at this point. More work needs to be done. You'll note that PFOS was detected at the highest concentrations, and that's typically what's seen in most PFAS studies globally. The sites with the highest levels were Hall Creek and Kings Landing. And then looking at the PFAS with the different biological endpoints. Only one, and I want to note too that this table only includes PFAS compounds that were not significantly different between the sexes. So these 3 compounds were not different between sexes, and so this includes all 50 of those fish. Size was only associated with PFNA, and generally that's also what we see in our other studies too. A lot of studies have reported that PFAS compounds are associated with liver and posterior kidney abnormalities, and we did see that with PFOS. Not so much PFDA. This is the bile duct myxozoan. This is a type of parasite. So, and hemolytic anemia is where we see a decrease in the number of red blood cells in the spleen. So it's interesting to find these results, especially with PFOS. As I pointed out earlier, arsenic was associated with hyaline generation in the posterior kidney, well, both of these posterior kidney endpoints. So it's interesting to see that we're having heavy metals and a PFAS compound having the same effects on the fish. So potentially having a synergistic effect, but this needs to be fleshed out a little bit more. Looking at correlations between PFAS and the different heavy metals. And what you'll note right away is that there's a lot of negative correlations, especially with copper. And we're not really sure why this is. Some of my collaborators on this study hypothesized that it could be competition for sorption sites within the tissues. Well, this is specifically the heavy metals were detected, analyzed from the muscle, and then the PFAS from the plasma. So the results here might look different if we analyzed these compounds from the same tissue, and that would be kind of interesting to tease out too. But arsenic is one that has been found to be positively associated with PFAS in earthworms. So that's something I kind of want to follow up on more. We're going to be doing more testing this year, and I'll see if this association holds true. So as I mentioned, we did also sample blue catfish last year, again from the Patuxent River. And I failed to mention early on that we started with the Patuxent River because it was the location where Maryland DNR has the most data available, specifically population data that our team here at the USGS is using for modeling. Last year, we also sampled 20 blue catfish from the Nanticoke River, and we will analyze them for the same endpoints and heavy metals and PFAS as we did at the Patuxent. These sites represented more commercial fisheries, and then this year we're going to be sampling sites where there's more recreational fishing that occurs, although there's overlap between those. This year we're going to be sampling at the Wicomico River in Salisbury, Maryland, due to PFAS contamination originating from the Purdue agribusiness facility. We'll also sample at the Anacostia River in DC, near the mouth of close to the Potomac River. And this is known to have a lot of legacy contaminants, urban runoff. I mean, it's surrounded by urbanization and combined sewer overflows. And then also we'll sample blue catfish from the Potomac River near Washington, DC at the Blue Plains wastewater treatment plant. I put this table up here. This is from the Maryland Department of the Environment, and this is their fish consumption advisories. And I just wanted to show why I was picking out the Anacostia River and the consumption advisories they have there in place for the Anacostia, Potomac, and Wicomico. At this point, most of the catfish consumption advisories are related to PCBs. And the reason I didn't initially start testing PCBs is because Maryland already has been doing that. I was trying to focus on things they haven't. And looking at plasma PFAS levels was related to the fact that Maryland wants to start using blue catfish for alternative reasons, not just human consumption. And they want to potentially use it as fertilizer or animal food, like in zoos. You'll note that there's no limit on the consumption of catfish from the Wicomico River, where there has been noted PFAS contamination. Anacostia, it's a little bit confusing here. I'm not sure where they sampled, but I mentioned earlier that size for some of these contaminants is not associated. For PCBs it is, but some of the other heavy metals and things. So I just want to follow up and see what the levels of the heavy metals are in these areas where they haven't been tested. So to conclude, heavy metal and PFAS levels were considered low when you compare them to other species. And I put in bold heavy metals, PFAS in the Patuxent River because I want to be explicit. This data is relevant just for these contaminants from the Patuxent River. Other contaminants could be a risk, like the tire degradate, 6-PBD-quinone. If you're going to eat catfish, blue catfish, I would urge you to consider the MDE consumption advisories, with the caveat being that if you're eating blue catfish from a restaurant or something that was collected by a commercial fishery, you won't know where it came from. And it's important to consider, again, that fish size was only associated with mercury and 2 PFAS compounds. Everything else that I tested, it was not associated with size. So these consumption advisories with size are not relevant for some of the contaminants I tested for. Inorganic arsenic, there is, like I said, there is consumption advisories set by the EPA sets a chronic oral reference dose for inorganic arsenic at 0.3 micrograms per kilogram of body weight for non-cancer health risks. But what they basically say is that you don't want to consume inorganic arsenic at all if you can avoid it. Maryland is pushing to put blue catfish in schools and other institutions. So this is something that should be followed up on. And microplastics were not detected in 5 blue catfish that were analyzed. I'm running out of time. So I just want to say that in 27, we're going to be doing a blue catfish reproductive study, following up on some work that was done previously in Virginia. So if anybody wants to follow up with me on that, please reach out. Here's some data that I have released on a different endpoint, the reproductive study. And here's my funding and acknowledgements. I'll just leave this slide up, and that's all I had for today. But mostly I do want to point out that the funding was provided by USGS Ecosystems Mission Area Environmental Health Program, and a lot of in-kind support by Maryland Department of Natural Resources.
That was a really excellent kind of dive into yet another aspect of how invasive species and human health intersect in terms of contaminants. So thanks so much for that great presentation.
Thank you.
But we'll move on. Yeah. Next up, we have Dr. Michaela Phillips. So Michaela, take it away.
Thank you so much. So we're going to switch gears a lot from what we've been talking about and kind of take a journey across the Pacific Ocean to Hawaii, which is where I live and work. And I'm going to be sharing some work in Hawaii, and a lot of the concepts that I'll talk about today are relevant across the Pacific, but just in the interest of time and kind of the way that I do my work, we'll focus on Hawaii. So we'll be talking about fire and invasive grasses primarily. So first, just to kind of orient you to the landscape, we're used to hearing about wildfire in the Western US, continental US, but not as much in Hawaii, although in 2023, the devastating wildfires on Maui really started to change that narrative. Those of us that live and work here have been saying that wildfire is a big threat for a long time, but it hadn't really made it into mainstream circles until the tragedy at Lahaina. This map is showing wildfires across the state from 1999 to 2022. And what's important here is that the warmer colors, the oranges, red, and black, are the more recent fires. If you look at Hawaii Island, which is the big island, this big one here, which happens to be where I live and where the work I'll talk about today is located, you can see that there are just the warmer polygons are much larger than, like, say, this little green one here. So what we're seeing is that wildfires are becoming larger and more frequent. You can see this same pattern I realize I'm like pointing with my mouse and you guys can't see that. You can see the same pattern on Maui as well. I wonder if I do a laser pointer. Maybe you can see that. So you can see the same pattern on Maui.
Yeah, we can see that.
Okay, great. Sorry about that. I have animations for the rest of it. So that map obviously showed that wildfire is increasing here. You can also see that burned area is increasing through time, with more than doubling between 1940 and 2022. And while that's happening, there's a lot of interest in what the cause is, since our native ecosystems are not used to wildfire and struggle to recover often after fire. So here we can see on the bottom figure, we have on the y-axis land under agricultural production, and we're going to focus on that blue line, specifically the areas with the red box around them. That is working pasture lands. So there was a huge increase in using lands throughout the state for cattle grazing in the early 1900s. And then that began to drop off after 1940, which is where you can see that significant increase in area burned. So before cattle were introduced, there was all native forest, which was then often removed to create pasture lands for grazing. And cattle ranching really became a big part of the culture in Hawaii, and still is in certain areas. But the amount of working lands have really degraded. So this map on the left in 1937, or really decreased. This map on the left, you can see all of that green. So if you look at just focusing on the Big Island again, there's, most of the state is this green color, which is land used for grazing. So over 2 million acres. And then when we move to 2015, we see that's dramatically decreased. There's less than half of the remaining working land, so just a little bit over 760,000 acres. And most of those lands, because they are no longer working pasture, the grasses that were introduced, mostly grasses, non-native grasses from Africa and other places, have really been able to reach high levels of biomass and don't have the suppression that came from grazing. And as many of you, I'm sure, know, wildfire can push these ecosystems into novel ecological states. So the photograph on the left here is a native forest in Hawaii, and on the right is a changed state to an ecosystem dominated by invasive grass. This invasive grass is called fountain grass. It's also quite an issue in Southern California if you've spent any time there. And it was actually brought over primarily as an ornamental originally here. And it's really taken over in the drier parts of the state. In the wetter areas, it's another grass called kikuyu grass is the common name. And that is a grass that reproduces using its rhizomes underground and can spread vegetatively, and it is from Africa, and it is a quality forage, so it was introduced for cattle grazing. So we'll see that these fires often come through these grass areas because they are more fire prone, and then they can carry into native forest areas. And then once a fire hits a native forest area, just like this ball here on the left, it can push an ecosystem past the threshold and change it to a novel ecological state that is dominated by grasses. So the grazing introduces grasses, but fire also begets more grasses, which can lead to more fragmented landscapes, smaller areas of native forest. The native forests take much longer to recuperate. So now we'll kind of focus in on a example of a fire that I've been focusing on a lot on Hawaii Island. So this fire is shown on the left in this red polygon. It's called the Leilani fire, and it burned roughly 16,000 acres on Hawaii Island in 2022, of August 2022. And this fire was ignited by military activities on the neighboring lands outlined in yellow. That's Pohakaloa training area. It's a military base. And then, so the fire started here and then spread into adjacent lands managed by the state. And of the area burned, about 2,500 acres contained montane dry forests and shrublands. And this was really tragic because this single event burned roughly 50% of the remaining montane dry forest in this area called Upper Pu'uanahulu. And just these polygons, the blue and the yellow on this map, there's an incredible amount of endangered plant species habitat within this area, and also a lot of dry forest. And dry forest is incredibly threatened throughout Hawaii, but also throughout the world. So this fire was really tragic. You can see the footprint of it in this photo. And on the right side of this photo, a lot of this sort of more golden vegetation is fountain grass. So the fire started on the base and then was able to really move very quickly because of all of that dry grass vegetation. So we've been trying to think about restoration for both this fire and just kind of the large scale needs throughout the state and on Hawaii Island. So one strategy we've been trying to develop here is aerial seeding. So aerial seeding is a scalable restoration strategy, and it's widely used in the continental US through programs like the Burned Area Emergency Response Program, also called BEAR. And it's also common in many other countries. But in Hawaii, this approach remains really underutilized because we just don't have the same capacity. We don't have, we have a lot of helicopters, but we don't have any seed spreaders on Hawaii Island. So many of these efforts do rely on hydroseeding, which you can see in that bottom right picture, but that's pretty impractical in the dry areas where I work at that fire that I just showed. And that's because bringing large amounts of water out to the field is just pretty impractical. So in response, land managers here have been trying to develop dry seeding mixes where we're dropping seeds from these seed spreaders and helicopters and other soil amendments to hopefully increase their success. And I've been collaborating with managers here to develop, refine, and monitor the outcomes of these dry seeding approaches. So now I'm going to talk about one of the pilot study experiments that we set up, and this was on the Leilani fire on the lands managed by the state. The photo in the top right is of Edith Adkins, who's the land manager there. So we set out to test seeding with seed alone and with mulch in an 18-acre area with 4.5-acre treatment blocks. We looked at 2 levels of mulch. You can see this photo here that's a blue dry mulch product. So we put seed down and also mulch, as well as, so 2 levels, a heavy mulch where there was a lot of mulch, a light mulch where there was less, and then seed alone. And this was to see if the mulch could help increase soil moisture, increase germination, and decrease erosion. So here, hopefully it plays, is a video of the mulch being applied. So this hopper that you see slung from the helicopter was actually brought over from Oahu, the island where Honolulu is. And while it worked for distributing the mulch, it didn't effectively or evenly distribute the seed. This is a photo showing how the mulch looked on the ground after application. And then you can see this little yellow spot here is actually a native seed. And so we had to think creatively because we couldn't use that hopper for the seeds. It was just running out very quickly. So the team ended up modifying an ATV seed spreader, which is what is slung here, to distribute the seed for the project. And this workaround was effective, in this condition, but it also highlighted the increasing need for seed delivery capacity on Hawaii Island to support future restoration efforts. And it just wasn't the most ideal way, and we weren't able to respond as quickly following the fire as we'd like to for future fires. So some results. First, we found that moss cover on the ground, so biological soil crusts, which are organisms of mosses, lichens, and lightly pigmented cyanobacteria that dominate soils in drylands worldwide, recovered really rapidly. So these organisms grow on the soil. You can see this photo in the middle. I'm holding what I like to call a moss cookie, and it's growing on the ash layer, and it's really keeping all of that soil together. So this was an encouraging to see this rapid recovery and see that there was mechanisms on the ground to decrease erosion and to hold soil moisture. We know biocrusts are great at keeping the place in place and decreasing erosion, increasing soil moisture. So that was quite heartening initially. And then here are some initial results. So you can see what the landscape looked like on the left after the seeding. And then on the right is a figure showing plant cover with native in this teal color and non-native in the purple color. So first thing, you'll notice the purple bars are smaller than the teal bars, which is good. And that is highlighting that the weed management approaches that we've been using, which was repeated herbicide treatment, are working. We're not actually seeing an incredible amount of native germination just yet. And part of that is we put these seeds out and then it was quite dry, which actually led to another kind of unexpected positive reason for using the dry seeding, is that the seeds can kind of wait until the conditions are right to germinate. Whereas if we did hydroseed, they might have germinated and then it got really dry and they would have had high mortality. But some of the we are seeing quite a bit of native regeneration once the invasive plants are managed, which is really exciting. And then you'll see these two red bars here. We're seeing a little bit less regeneration under this heavy mulch. And part of that, so we're seeing that might be limiting, the heavy mulch might be limiting both natural regeneration and seedling establishment from the aerial seeding. And this site is dominated by pahoihoi, which is the Hawaiian word for smooth, hardened lava, which you can see shown in this photo here. And it's an uneven lava surface, and there are a lot of cracks. So we think that that it caused the mulch to accumulate much thicker than intended in these little pockets where there is soil. So this is kind of making us think maybe the mulch isn't the best approach. So because this study raised so many new questions and underscored the need to improve seed delivery methods, we wrote a proposal to the Environmental Security Technology Certification Program, and we're now launching the project. So the first part is really exciting. We're buying a seed spreader that will live on Hawaii Island and belong to the state, to the Department of Forestry and Wildlife, and be a resource for the whole island to use for partner agencies. So our first step is calibrating it through test flights, and then we're going to create a user manual so it can be used. Next, we're going to run field trials on the Leilani fire, lands impacted by the Leilani fire. So both on the military base as well as on the state lands. And we'll use the hopper to test treatments, seed alone. And then we're actually not going to do the mulch plus seed, and we're exploring using seed coating technologies in lieu of that. But we will look at the application of biocrust moss that we're collecting and propagating on site and seed to see if we can kind of have this nature-based solution for erosion control by introducing and supporting moss establishment. And this moss treatment kind of leverages the propagated biocrust to improve both seed success and erosion control. And then across sites, we'll track community responses, biocrust cover, and soil conditions. And then lastly, we have a really robust communication component where we'll share results through workshops on island and throughout the state, and focus on the protocols for using the hopper, lessons that we learned from the fieldwork, as well as collaborative planning for advancing this technology. And this project is really highly collaborative. It's co-developed with the Forest Service, the state, DOFA, as we call them here, and Pohakaloa training area. It's been really fun to work on. So it's been a little bit slow to start because of just challenges with adjusting to administrative changes and moving money and changes in staffing. But we have purchased the seed spreader, and right now we're collecting and propagating biocrust and also collecting seeds, and we're hoping to be doing the seeding next winter. So this project really begins to tackle a critical gap. Like, once we have more seed, how do we effectively deliver it across landscapes in ways that can support successful establishment? And part of this is because there are parallel efforts to really increase seed production in Hawaii. Most restoration efforts are so far done with outplanting seedlings on the landscape, and it's just not scalable to what is needed following fire. So I just want to take 1 second we've been talking primarily about dry forests, but wildfire is a major concern for dry ecosystems in Hawaii, and it was historically less of an issue in wet forests. However, with increasing drought and a long legacy of land use change in these systems, fire is becoming a growing threat across ecosystems, including those wetter forests that were once considered relatively safe. So this is footage from the Mono Road fire, which just happened in November 2025. It burned roughly 2,100 acres on the slopes of Mauna Kea, and it's right alongside Hakalau Forest National Wildlife Refuge, which is fortunately, it didn't cross into the refuge. The refuge is an incredibly important place for threatened and endangered native Hawaiian forest birds. And it's just a really beautiful, special place to those of us here, and also many people that have visited. And what's interesting about this fire is, while there was grass at play, it was dominantly a gorse fire. So you can see gorse photographed here. And in addition to the Mono Road fire, there was another fire near the refuge in September 2025. The Ka'ala fire was relatively small and burned about 523 acres. But together, these fires highlight the risk posed by a dense grass understory and this dominant gorse within the refuge, which can easily carry fire and threaten both restored and native forest areas. Gorse management is a huge challenge up in this area. And so there's a group of us that are trying to take this fire as an opportunity to do some native forest restoration and other restoration efforts to try to knock back the gorse while we can, because it's a really tenacious plant and also burns quite hot. So much different than the grass fire cycle that I've mostly been talking about, but something I wanted to touch on as another big challenge in Hawaii. So I just want to end with discussing some of the fuels management approaches that are used here and there's interest in. So fuels reduction is a big thing. There's a ton of herbicide application, as well as weed whacking alongside roadsides and things like that, and even interest in using grazing for fuels management. A lot of these big land management agencies like Pohakaloa training area and the state create huge fuel breaks alongside their roads, and are also using native forest restoration to create these vegetative fuel breaks or green breaks. to hopefully decrease fire risk, though it is an uphill battle with winds. And then native forest restoration is a key tool for decreasing fire risk, but scaling up is a really big challenge. So traditional grass removal and outplanting trees is really expensive. And the estimates that we have from this recent paper, or it's not that recent anymore, past 10 years, range from $5,500 per hectare to more than $13,000 per hectare. So it's really a long-term strategy. And that's one of the reasons we're really trying to increase seed-based restoration efforts to be able to cover more land at a cheaper, at a much cheaper cost. Fuel and fire breaks are really commonly used tools here. The upfront costs are lower than restoration, but they require a lot of maintenance, and there's a lot of interest in what width is needed, and there's a need for research there. The Leilani fire was able to jump the road just because it was so windy. And then the last thing I just want to touch on is that there is a lot of work happening on the ground and different great groups working on wildfire as a shared responsibility and bringing community into these conversations. And I didn't really talk about that today, but I wanted to acknowledge it's happening. So there's the Hawaii Wildfire Management Organization, and they produce wildfire protection plans. This is just an example of one for South Kona, as well as the Pacific Fire Exchange. And this is showing a map of burned areas across Hawaii Island. So these communities are really working within, or these organizations are really working within the wildland urban interface and beyond to make sure that that our population centers that are at risk of wildfire are more prepared and are part of conversations with planning. And with that, I'll wrap up, and I don't think I have time for questions, but maybe I can take some at the end.
Yeah, thanks, Michaela. We do need to move on, but that was really excellent. Encouraging to see some of these new approaches to help address this tricky fire and invasive plant cycle. We'll go ahead and hand off to Dr. Mary Beth Bray to do our final presentation, and your slides are up and looking good, Mary Beth, so
Excellent.
take it away.
So I'm going to talk today about how we've developed acoustic deterrents for invasive carp control. So going into the fish realm. And I do want to acknowledge a whole lot of people. So I have sort of the honor of representing a very large group of people. And so in the left-hand corner here is Dr. Krista Woodley. So she's my partner in crime at the U.S. Army Engineer Research and Development Center. And then this whole top row has helped significantly with a lot of the analyses that you'll see, and then our very large field teams to do almost 10 years worth of work now. You guys still see my screen? Are we good? Mine bounced.
All good. Yep.
Thank you. All right, so let's jump into this. I'm going to give you or take you on a river journey today. We're going to start out up here with invasive carp, and I'll tell you what they are and what we're trying to do about them. Next, we're going to talk about a little bit about their life cycle and controls. Where do we find them? And then why are we working on acoustics to control them? A little bit about our research and development process, how we go from an idea to a management tool. A little bit about locks and dams, which are fish highways and toll booths. And then a lot of time on this underwater acoustic deterrent system that we've been developing for almost 10 years now, and a prototype study on a navigation lock on the Mississippi River. If that's not enough, I'll talk a tiny bit about technology transfer. So that's where we're headed. If you're not familiar with invasive carp, there are 4 species: silver, bighead, grass, and black carp. These are also referred to as the 4 Chinese aquaculture carps or invasive carp of North America. They're originally brought from Asia to the US in the late 1960s, early 1970s to control algae in wastewater treatment facilities and ponds. They escaped containment in about the 1970s, and they've slowly spread, slowly initially, not anymore, and continued to expand their range throughout the Mississippi River Basin. The map that you see on the right-hand side is the silver carp distribution as of February 7th from the NASS database that USGS maintains. So there's various control measures that USGS has been working on, as well as others, to control these fish species. And a lot of these control measures are conducted at large locks and dams. And so what I'll show you in the end is how we use those lock and dam structures to control these fish, and how we use one of the mechanisms, which is underwater sound. So before I go into sound, I need to explain a little bit about the carp life cycle. And so you can see that generally here. And so you have fish, females and then males, that move into a spawning habitat. This is often in a main channel site somewhere with a lot of flow. They're broadcast spawners, so the eggs go out and then milk from males will be dispersed. That drifts downstream, eggs are fertilized, eggs drift through this egg drift corridor and are kept up in the water column. There's that's the larval drift corridor. So as the fish develops, it continues to move downstream and then into some juvenile habitat, typically backwaters or slow flow areas. And then the adult habitat, which can be anywhere in the river, and that kind of changes throughout the year. And then they move between those habitats and eventually up into uninvaded areas. And so we can't control fish equally throughout this entire life cycle. Can use contract fishing and removals in this area and the uninvaded areas and these movement corridors. We can use things like chemical controls to control juveniles or maybe eggs. Can manipulate habitat, so drawdowns of water. Can try predator enhancements or attractants. And then we have deterrents or barriers, and these can be applied in some cases at juvenile habitat level, but mostly we're talking about this area right here as fish move between adult habitat, spawning habitat, and uninvaded areas. And so this is the area that I work. Also, why do we care? Well, if you haven't seen or you haven't been to the Midwest US, this is a video from the Illinois River. And it actually highlights why underwater sound could be useful for controlling these fish. And so this is their response as a boat goes by or they hear a boat motor. So we knew already that they responded this way when boats went by. And so we thought, what's the Achilles heel of these fish? How can we use that? And so, oops, I don't need to see it again. We thought maybe we can use this underwater sound to deter them. Now, how do you learn what a fish can hear? We can't put a fish into a audiology booth and ask it, to press a button when it hears a sound. So what we did initially is we measured a couple things. We measured sound pressure and we measured particle motion. So these are the two components of sound. So the graph on the left, well, both of them have frequency on the x-axis. And then the two components of this traveling energy wave that we know is sound. For 5 native Mississippi River Basin species and then 2 invasive carps. So the 2 carps are in these brighter blue and pink colors here. And carp have this Weberian apparatus. So this is their swim bladder. It's actually connected to their inner ear. And so they sense or feel the sound, hear the sound, oftentimes more intensely than native species do. And so we looked at these 2 components of sound. And what we saw was that excuse me, carp are more sensitive to sound pressure and particle motion at nearly all frequencies compared to most native fish tested, and that their hearing range is generally higher than most fish up here. And so you can use this information to kind of take advantage of it and apply unique underwater sounds that hopefully are outside of native fish hearing range and target more closely the bighead and silver carps. And so we take this research and development approach. We start with lab studies, and I showed you these hearing tests. We kind of do proof of concept testing. We move into a little bit larger areas, into pond studies. We do more acoustic monitoring, signal development. Then we move into the field. So does this actually work in field studies? And then what I'll show you at the end here are these long-term studies at navigation locks, and then moving into a transfer of the technology. And so we go through this process with the goal of developing an underwater acoustic deterrent system that alters invasive carp behavior in a predictable manner. So we don't want these just going anywhere, but also limiting the impacts to native fishes. So that's our big goal. Easy. We also have to take into consideration this life history and this behavior of the fish. So knowing when they move, where they're moving to, and where we can best control them. So we started with the lab studies. We moved into these pond and field studies. These are just some examples of the work that we have done. These are the ponds here in La Crosse, Wisconsin, trying to move fish back and forth between these two areas with sound. This image on the right, it's actually a video. This off. And sound will be played in this left culvert, and you'll be able to see exactly when the sound is turned on here. We call this carp popcorn. Okay, so we were trying to see if you could exclude fish from one side or the other by turning sound on or off. So this was a study done in the Illinois River. And then we've also moved into these areas like the Army Engineer Research and Development Center, or into the field to look at different sounds in the lab, and then transitioning those to smaller field studies. So once we've done that, so we've moved down here into these field studies, we have to say, all right, how does this work at a large scale? Can we really do this at a place where we need to control them? And so we took this work to Loch 19. This is a study site on the Mississippi River, located in this star here. And this is the site. So here's the lock approach channel. This is actually the installation of the deterrent. It's built across the Des Moines Rapids, so there's a huge dam extending across here. And the unique thing about this site is that this is Loch 19 right here. So this is the percent of days the river is in open river conditions, which means that all the gates are open, and the tailwater down here is equal to the pool level on the upstream side. So fish can swim through just like an open highway. And the one thing you'll notice is there's a 0 for Loch 19. And so at this site, this is fish movements already controlled exclusively through the lock approach and into the lock. And we don't have to worry about a deterrent over on this other side. And so with our state and federal partners, we identified this site, came up with some objectives to install and design and install an underwater acoustic deterrent system, evaluate the effectiveness at full upstream passage, which is a management outcome that people were interested in, assess the more fine scale behavior of these fish and native fish with the deterrent on and off, and then evaluate the operational performance over time. So here's a site layout. This map here has the lock approach channel on the left-hand side. So from the image that you saw, we were looking up into or towards the lock. So you'd be coming, if a vessel were moving upstream, it would move from left to right. Flow moves downstream from right to left. So you have the lock approach channel. The deterrent was installed in this last downstream discharge lateral. There's the sound bar, as we refer to it. And it was recessed into this lateral so that it's out of the way of vessels and navigation traffic. And we were lucky to be able to do that with a great design team. The discharge here, so when the lock empties, they have to discharge the water when it comes down. And it discharges here in this area in the downstream approach, as well as in the tailwater, which is over here, and that's on the closer on the dam side. So you have two areas where the water discharges. The approach itself is over 500 feet long, and this is a 1,200 foot lock, which is the largest on the upper Mississippi River. So vessels don't have to break in half to pass. So we installed this deterrent here. And then you can see these darker colors. So the dark red here is where the deterrent is. This is going to be the loudest area. This is the loss in decibel levels from the sound source, the red, and moving downstream and upstream. As it gets more purple or blue, that means it's quieter. So as we're down here in this dark blue color, sound is back to basically background by the end of the lock approach channel. So we create this gradient of sound. At this location, as I mentioned, fish can only move through the lock, so we're not worried about passage through the dam and monitoring that. For purposes of this study, we had this sound running 80 hours on and then 80 hours off, so we could test whether there were differences between on and off periods over different environmental and operational conditions. Okay, so how do we monitor fish? We monitor sound, we're developing these sounds. How do you monitor fish at this large scale? So here's an overview of the site. This yellow rectangle is the lock and lock approach channel, so our study site. And the star down below is actually where we released fish, so note that. We had 2 different acoustic telemetry systems. So this is a really fun part of my job is we get to put these little transmitters in these fish. We sew them up with sutures. These transmitters ping, and then they're picked up by these receivers that we have located throughout the entire area. With some of these receivers, we can position the fish just like you're positioned in your car or you're walking. And so we can create these kind of spatial maps and see how fish move throughout the area. And so that's how we track them. We had 2 focal species. We tagged more of invasive silver carp and native bigmouth buffalo. Here's a list of all the species that we tagged. So the ones highlighted here are the focal species. So we tried to get enough of those tagged over 4 years. And then we captured them actually most above Lock 19, upstream from Lock 19, and that's because they showed a greater propensity to try and move back through the lock if they had already been above the lock. And so we translocated quite a few of the fish. And then released them downstream, almost all of them down at this star, so about a mile downstream from the site. All right, so I'm going to jump right into results. One of the main items of interest was this full upstream passage of carp. So I'm only going to show right here the species that had, I guess, larger numbers of passage, ones that we can put into models and get statistical significance for. We had a lot more. If you're interested in all the other species, let me know. I'm just going to show a subset here. So full upstream passage means a fish had to move from the downstream pool into the lock approach, into the lock, and all the way upstream into the next upstream pool. And so what we saw for silver carp was fairly low numbers. This is a low passage site anyway, but we saw a 61% decline in silver carp passage when the system was on compared to when it was off. For bighead carp, closer to 0% change, actually a little bit more upstream passage. And grass carp, very few of them, but a decline, 75% from off to on. Native species, very close to 0 for bigmouth buffalo, a very small 5% difference in upstream passage. Paddlefish showed more upstream passage when sound was on. White bass less, and freshwater drum, there was no change. Now, movement into the lock, we had a subset of the data that we were able to use to look at, okay, so if they didn't make full upstream passage, were they able to get past the deterrent and into the lock? And how many did, and was there a change here? And we saw consistent results with silver carp. So 46% decline in movement past the UADs and into the lock when sound was on. Bighead carp, 38% decrease, increase for grass carp, and then fewer passed upstream when sound was on for bigmouth buffalo. You can see the other numbers. And so we haven't run statistical significance testing on this. That should be done very, very soon. The crew's working hard on it to determine if these results are significant or not, but these are preliminary results. We also looked at changes in the rates of fish movement. So I showed you the lock approach channel and the lock. What we did is we started with these general zones, and we're interested in what's the rate of movement over time, and how does the deterrent change that rate of movement. So we started with these broad areas and identified zones, and then we moved and over and overlaid this sound map on top of it. And then we created smaller zones, so a UAD zone, mid approach zone, downstream approach zone, based on the sound pressure levels and the sound we projected, and then looked at changes in movement rates with different conditions. Just going to show you the differences using sound off versus or on versus off here, but we're looking at a lot of other covariates as well. Okay, so this is just flipping that on its side. Here's our kind of broad areas, zones that we're looking at. And so moving to the right would be a fish advancing towards the lock and the deterrent. Arrows to the left means they're moving or retreating away. And so what we saw is compared when fish were moving, advancing, they were 16% slower to move from the pool outside the lock approach into the approach channel when sound was on compared to off, and then 35% slower to move from anywhere in that pool or approach channel into the lock chamber when sound was on versus off. We didn't see any significant changes moving from the general area out into pool 20 or anywhere, so that was not significant. When we look at the smaller zones, we saw a 16% decrease in the rate of movement over time into the middle zone, and then another 8% decrease into the UAD zone. And then 35% faster to retreat. So movement from the UAD zone and then the middle approach zone, 13%, but not back out into pool 20. So generally upstream movements towards and into the lock were slower when the UADs was on. Movements away from that immediate location of the UADs were faster when the UADs was on. If we look at other fish that had significant numbers to test, we see that bighead carp, grass carp, and bigmouth buffalo showed no significant difference in their rates of movement when the system was on compared to off at this broad scale. But when we look at this at a little bit finer scale, we see more localized changes in movement. So I'm only showing the significant changes here. For bighead carp, we saw that they were faster to retreat from the UADs and middle approach zones when sound was on compared to off. Grass carp were slower to advance into this kind of mid zone and faster to retreat from the UAD zone. And bigmouth buffalo were 12% slower to approach into the UAD zone, but no other significant differences. So no retreat differences. Finally, on fine scale behavior, we looked at, just again showing the significant changes here, we looked at every species. These were the ones that showed a significant difference in turnaround distance from the gate. So if the sound's on, we were interested in seeing, did they only approach so far and then turn around? How far is the sound affecting them from the gates? And so up here where 0 is, this would be the lock gates. And as you move down in this graph, that's moving downstream. So from bottom to top is moving upstream. The UADs is located at this red dashed line here. And so we show these violin plots to show the distribution of where fish turn around when sound is off on the left-hand side, right, or on the right-hand side. So for silver carp, we saw that they turned around probably 38 or so meters downstream from the deterrent when it was off. And when it was on, that shifted downstream 7.1 meters, and that was significant change. For bighead carp, they shifted their turnaround distance almost 4 meters downstream, so a little bit less. And then paddlefish actually showed a significant difference in turnaround distance, 11.4 meters downstream. Okay, so that was a lot. So try and summarize some of the fish movement data here. What we saw is consistent results for silver carp. So this is really good, very promising. We see an impact of the UADs on silver carp. They turn around further downstream. They advance slower from all the zones. They retreat faster from the UAD zone and the middle approach zone of the lock approach. and passages, both these full passages upstream and just passages into the loch, were decreased. We did see some impacts likely to bigmouth buffalo. We're still working on the significance testing here, but it is possible that there's some impact, and we'll have to work with managers to say what's the tolerance level of using a deterrent and having effects on native species versus carp. Same for bighead carp, grass carp, and the other native species. We had a lot of mixed results. And so we're looking at additional analyses and significance testing to kind of parse out what's going on here. And so what are we doing next? So we completed 4 years of this study, developed an acoustic deterrent, designed it, installed it, tested it for 4 years. We had a 5th year of operation this last year, and we will have one more year this coming year where we have the sound on 100% of the time. And so we want to see what's the change if we don't have these on-off conditions, and we're only looking mostly at fish passage, not these fine scale behaviors for these years. We're going to continue to look at the analyses, refine these a little bit that we've been working on, get significance testing, and then we're working with management agencies, states and federal partners to kind of transfer this technology, say what have we missed? And how do we do that? So we've been very fortunate to have a great relationship with all of our state and federal partners on this. At the bottom here, we have 2 patents. Well, one patent has been approved for the sound bar, that giant thing that you see swinging on the bottom here. So that is patented. And the engineered signals that we use that are developed by ERDIC, those are patent pending, but we can use those. We also have been on a number, worked on a number of project design kind of panels as experts to provide technology transfer information. So what we've learned here has been used as the foundation for the Brandon Road Interbasin project. So this is a multi-deterrent that is being designed and installed at Brandon Road Lock and Dam to prevent carp moving into the Great Lakes basin. And so the acoustic deterrent part of that, we've transferred everything that we've learned here to them, equipment, operations, methods, ways to cost save, the communication system, limitations, considerations, all of that. We've also helped with the providing information for the Section 509 process to install deterrents in the Tennessee-Cumberland-Tombigbee waterway, and helped with informing the design of an acoustic deterrent that's going to be installed at Lock and Dam 5 on the upper Mississippi River. So we're just continuing to work with partners to say what's the tolerance levels, do we need to do any more research, or how can we help transfer this technology into a management application. Here's our downstream lock approach of 19, where I've spent a lot of time on a sunny day. And then a bunch of great pictures. This has been a huge, huge effort. Many partners, funding as possible from the Great Lakes Restoration Initiative because of the need and applicability to the Great Lakes, and also funded by the USGS Ecosystems Mission Area, which supplied substantial invasive carp funding through the project through annual appropriations. And then in-kind support from many of our upper Mississippi River invasive carp partners, both state and federal. So if there's time, I would be happy to take any questions, or you can email me here if you have anything you'd like to know. Thanks.
Thanks so much, Mary Beth. And we are just at the top of the hour, so we'll probably need to close out. But yeah, thanks for your email address. Big round of applause for Mary Beth and everyone. It's great to see those virtual hands going up. I'm going to hand things back off to Tegan at NISAW, but just wanted again to thank all of our stellar presenters from USGS. Thanks to the NISAW team for hosting today, and of course, thanks to all of you for joining in. I will put in here a link to some information and email addresses for a few of us if you have follow-up questions on any of these presentations or USGS invasive species science in general. We'd love to hear from you, and best wishes to all for a continued excellent National Invasive Species Awareness Week.
Thank you so much, Paul, and thank you so much to all of our speakers. It's been a great way to kick off National Invasive Species Awareness Week. And so here at NISMA, we do like to end our information sharing sessions with a call to action. And today we'd like to ask you to send a letter to your federal representatives in the House and Senate asking to support the Mississippi River Basin Fishery Commission Act. The bill would establish a coordinated federal state commission to manage and restore fishery resources across the Mississippi River Basin. You can either scan the QR code or follow the link in the chat. Input your information and the system will automatically select your senator or representative and send him or her the letter we have prepared. We have a range of bills included in that letter. And lastly, I just want to show you the schedule again. We have a great lineup of webinars scheduled for this week. You can visit our NISAW landing page or our events page to register. We look forward to seeing you throughout this week as we continue to celebrate NISAW. And as a reminder, we'll be posting all of our webinars this week on NASMA's YouTube channel. So if you missed any part of today or aren't able to attend some this week, please do check those out later next week. We've got some of those emails coming in the chat, so hopefully you can follow up with folks there. But again, just want to thank everybody so much for joining us.